Okay, in this video we're going to look at how we can target proteins to the mitochondria. As we now know, protein production starts in the cytosol of the cell, and therefore there must be a way of targeting proteins that are required by the mitochondria to the mitochondria. The mitochondrion is a fascinating organelle. For a start, it has a double membrane. It also has its own DNA and can make its own proteins. However, it is reliant upon the cell for certain proteins, and these proteins are made in the cytosol and then trafficked to the mitochondria. So how does the cell know which proteins to send to the mitochondria, and how do those proteins get into the mitochondria? Well, it's back to the amino acid sequence providing a signal peptide that acts as a postcode that tells the cell that the protein should be targeted to the mitochondria. In this case, the precursor proteins have an N-terminal signal sequence that says the protein should be imported into the mitochondrial matrix. That is, the protein should be trafficked across the two membranes to the mitochondrion interior. And once the protein is imported, a signal peptidase will remove the peptide address at the N-terminal. The sequence you can see to my left is the mitochondrial targeting sequence, and this has some interesting features, in that it folds to give an alpha helix that has positive amino acids on one face and non-polar on the other. And it is this alpha helix that identifies the protein as mitochondrial, and the helix is recognized by a receptor on the mitochondria. Now, mitochondria have a double membrane, so how do we get proteins across? And how do we get proteins that need to be between the membranes to that location? Well, the process comes down to a series of pore proteins in the mitochondrial membrane. And these proteins can be divided into the TOM complex, the translocase of the outer membrane, and the TIM complex, the translocase of the inner membrane. And TIM and TOM are responsible for passing the protein from the cytosol to the correct location in the mitochondria. There is also a third protein complex, the OXA complex, which sits in the inner membrane of the mitochondria and transports proteins that are made in the mitochondria or have been imported into the matrix of the mitochondria to the intermembrane space between the outer and inner membranes. The TOM complex is an example of a gated pore. That is, the complex contains a receptor for the signaling motif on the protein. And once the protein is bound to TOM, the protein is then threaded through the pore of TOM and through an aligned pore of TIM into the interior, the matrix of the mitochondrion. However, this process can get a bit complicated in that sometimes the protein will not thread all the way through the TIM into the matrix, but will stop due to the presence of a stop transfer sequence. Hence, this means that we end up with the majority of the protein in the intermembrane space. Sometimes a protein may make it to the matrix of the mitochondrion only to be recognized by OXA as an intermembrane protein and sent back. Once a protein has been sent back to the intermembrane space, it can either remain attached to the inner membrane or be cleaved to become free-floating in the space. It is also possible to produce proteins that thread backwards and forwards across the inner membrane. To achieve this, you need a series of motifs and some chaperones. Okay, so that is it for getting proteins into the mitochondria.